that your engagement will make this session truly interactive and insightful. Please feel free to share your questions and comments using the chat feature. We will address as many questions as possible during the question and answer session. Today's webinar will focus on key aspects of biomanufacturing of monoclonal antibodies from both academia and industrial perspective. This platform aims to foster valuable insights into this critical subsector, advancing collaboration across academia and industry. We are delighted to have an esteemed panel of speakers who will share their expertise with us. From DBT, we have Dr. Vaishali Punjabi, Scientist F DBT, and Dr. Vashniya Singh, Scientist D at DBT. Academic perspective will be shared by Dr. Anmol Chandele, ICGB, New Delhi, and the industry perspective will be shared by Dr. Vinay Konaje, Navya Therapeutics Private Limited. Without taking more time to commence this session, I invite Dr. Vaishali Punjabi, Scientist F DBT, to set the context for today's session. Over to you, ma'am.
A very good afternoon to all of you. On behalf of the Department of Biotechnology and BioRac, I welcome you all to the 7th webinar on biomanufacturing of monoclonal antibodies under the Biomanufacturing Initiative of the Department of Biotechnology. The convergence of biological sciences with advancements in computing, automation, AI, and data analysis is sparking a biorevolution with the potential to transform economies and societies. This revolution could revive the industrial revolution in its disruptive impact, influencing areas like food, fuels, materials, and healthcare. India is also experiencing rapid growth in biotechnology industry, with its bioeconomy expanding 13-fold from $10 billion in 2014 to over $130 billion in 2024. It is projected that India's bioeconomy will reach $300 billion by 2030. Department of Biotechnology and BioRac have played a pivotal role in nurturing an ecosystem for boosting India's bioeconomy through its various schemes and programs. Through these efforts, India is now poised to join a new global wave of industrial revolution, focusing on more sustainable manufacturing practices driven by recent advances in biotechnology. Recognizing the national potential of biomanufacturing to drive India's bioeconomy, the Union Cabinet, under the chairmanship of Honorable Prime Minister in August 2024, approved the Bio-E3 policy for fostering high-performance biomanufacturing in the country. Subsequently, the Union Cabinet has approved the umbrella scheme of the department, Biotechnology Research, Innovation and Entrepreneurship Development, BioRide, on 18th of September 2024. And Biofoundry and Biomanufacturing is a new component under this scheme. Through extensive national consultations, 6 thematic sectors have been identified for the Biomanufacturing Initiative. These are bio-based chemicals, enzymes, smart proteins, functional foods, precision biotherapeutics, climate resilient agriculture, carbon capture and its utilization, and marine and space research. A strategic roadmap has been formulated to implement these thematic verticals under the Biomanufacturing Initiative. Call for inviting proposals under these thematic verticals will be announced soon. Regulatory conclaves are being organized to develop framework for coordinating regulatory reforms for each of these thematic verticals. The policy also envisions setting up of bioenablers such as bioartificial intelligence hubs, biofoundries and biomanufacturing hubs to support innovation and scale up across the thematic verticals. The first DBT BioRac joint call for proposal titled Mulankur Bioenabler, Biofoundries and Biomanufacturing Hubs was issued, and a large number of proposals were received under this call. India's first biomanufacturing institute, the National Agri-Food and Biomanufacturing Institute in Mohali, was inaugurated and dedicated to the nation in October 2024. A memorandum of understanding has also been signed between DBT and ISRO for cooperation in space biotechnology and biomanufacturing in October last year. We are already also in discussions with the Ministry of Earth Science to formulate an MOU for setting up of Advanced Center for Marine Resources in the country. Recently, a center-state partnership conclave on the Bio-E3 policy chaired by the Honorable Minister Dr. Jitendra Singh Ji was organized on 7th of February 2025. Discussions were held to facilitate collaboration between the center and the state governments under the Bio-E3 policy with representatives from 25 state science and technology councils, industry invitees, senior officials from both DBT and BARAC. During the conclave, state governments were urged to set up Bio-E3 cells that will serve as an interconnected knowledge hub linking states and national stakeholders. Outreach activities for Bio-E3 policy are being carried out extensively through press meets, social media platforms, editorials, school campaigns, etc. A website for the Bio-E3 policy has also been launched. To further disseminate information about the Bio-E3 policy and discuss the various thematic verticals, DBT and BioRack have launched the webinar series on Biofoundry and Biomanufacturing Initiative in November 2024. Today is the seventh webinar under this series. Today's webinar focuses on monoclonal antibodies, a transformative vertical under the Bio-E3 policy, highlighting the latest advancements and their critical role in shaping the future of sustainable biomanufacturing in India. Monoclonal antibodies represent a promising avenue as the first line of prophylactic therapeutic options during outbreaks, pandemics, and also as complementary approaches where vaccines cannot reach, such as very young, elderly, or immune compromised individuals with vaccine or individuals with vaccine hesitancy. DBT envisions developing a biomanufacturing ecosystem that can promote development and manufacturing of monoclonal antibodies to improve patient access to cost-effective and high-quality monoclonal antibodies, become a significant global hub for technology leadership and biomanufacturing, build capacity to address ongoing health needs, and respond to emergencies. Once again, I warmly welcome you to today's webinar and hope you find the discussions insightful and inspiring. Thank you very much.
Thank you, ma'am, for providing an excellent overview of the vision and objectives of this initiative. Our next speaker is Dr. Vasne Singh, Scientist D at DBT, who will discuss briefly about biomanufacturing of monoclonal antibodies from DBT perspective. Over to Dr. Vasne Singh.
Thank you, Dr. Amit. I'm Dr. Bashne Singh from the Department of Biotechnology, and over the next few minutes, I will provide a primer about this sector, which is monoclonal antibodies. Can I have the next slide, please? So, as you all know, that monoclonal antibodies is a class of precision biotherapeutic, and this leads the global biopharmaceutical market. As you can see from the numbers, that the market was around $75 billion in 2013, and over the past decade has grown up to around $250 billion, with projections expecting a further increase to beyond $800 billion by the year 2032. More than 160 monoclonal antibodies have received regulatory approvals and are available in the market globally. And India also has a robust antibody market and is producing roughly 20% of these antibodies as biosimilars. And 4 novel monoclonal antibodies have also been manufactured by Indian manufacturers. The Indian monoclonal antibody market is pegged at around $210 in the year 2024. So the monoclonal antibody product development cycle involves several steps, and the Department of Biotechnology over the past few years through various schemes and programs, especially the National Biopharma Mission, has tried to address some of these steps in an integrated fashion. The steps involved beginning with discovery and early development, followed by process development and scale up, analytical characterization and cell line banking, and CAGMP manufacturing unit for clinical batches, followed by phase 2, 3 clinical trials, eventually resulting in market authorization. So, as has already been mentioned, the Bio 3 policy was approved by the Union Cabinet in the month of August 2024, and six thematic sectors had been identified under this policy, with precision biotherapeutics being one of these. And monoclonal antibodies development is being supported under this particular vertical by the Department of Biotechnology and Bio. So a SWOT analysis of the sector was performed. And as you can see from the slide that several strengths exist within the country for this particular sector, which includes a robust and growing market for monoclonal antibodies, availability of large-scale manufacturing expertise within the country, and availability of trained human resources. However, the sector also has certain weaknesses, which includes limited availability in public health settings, the high cost issues associated with monoclonal antibodies, low process efficiencies, and the biosimilar market is restricted to mostly domestic supply and has not been able to capture the global market as such. Also, several opportunities exist within this sector with global advancement in various antibody formats and introduction of innovative manufacturing technologies such as continuous manufacturing and PAT. There are also certain threats for this particular sector, which includes dependency on the global supply chain for raw materials involved in the manufacturing process, limited regulatory guidance pathway for novel antibodies, and global competition from other countries within this sector. So, to address this particular sector, the department has a vision of developing a biomanufacturing ecosystem that can promote development and manufacturing of monoclonal antibodies, improve patient access to cost-effective and high-quality monoclonal antibodies, become a significant global hub for technology leadership and biomanufacturing, and to simultaneously build capacity to address ongoing health needs and respond to emergencies as they emerge. So, for addressing all these factors, the department will be focusing its efforts on two lines, which is supporting discovery and application oriented integrated network research and bridging the gaps for scale up for the biomanufacturing sector. Thank you.
Thank you, Dr. Vashni Singh. Now it is my pleasure to welcome Dr. Anmol Chandele, ICGEB New Delhi, to present the academic perspective. Dr. Anmol is a group leader at the International Center for Genetic Engineering and Biotechnology, New Delhi. Her lab has been at the forefront of pioneer discoveries in human monoclonal antibodies, advancing their generation and characterization to deepen our understanding of B cell responses. A compelling outcome of this work has been the identification of potent neutralizing monoclonal antibodies, offering promising avenues for immunotherapeutic solutions, particularly for infections which lack universal vaccines. Dr. Chandeli, the floor is yours.
Thank you, Dr. Amit. Thank you so much for having me. It's really fantastic to hear from Dr. Vaishali about all the things that DBT is going to do. So labs like mine are super excited. So what I'll do for the next 9-10 minutes is that I'll just discuss what my lab does in the sphere of discovery. And so we get an understanding of what is going on towards the fundamental biology side. And I hope there are some students that can take some clues from it. So could I please have the next slide? So this is a very simplified summary of the way my lab functions in the sphere of monoclonal antibody work. My lab is a basic human immunology lab. We are interested in looking at B cell biology, as Dr. Amit has already said so. But one fantastic outcome of it is that we make monoclonal antibodies to look at B cell biology. And some of these can now be channeled towards these fantastic opportunities that DBT is creating. So if you look at this slide, on the left side, if you focus on the discovery part of this slide, my lab, if I can say it myself, is an expert on the left side of this slide. So we have fantastic systems in place where we can assemble human samples, we can characterize them, we can put them together in terms of the fundamental assays that we need, gauge their immune responses, bait the B cells, and also make the monoclonals. So all that left side is, very well set up. Where we are just moving into is the perfect opportunity that DBT is also doing this, is the part of development. So, the in vivo protection, stabilizing clones, process manufacturing, moving further is something that my lab is just in its infancy and is starting off. So this is actually a fantastic time for assistance, collaborations, networking, and talking so that we can move all this further and accelerate it. So can I please have the next slide? So here's how we do it, and I thought I'll spend a few minutes just because there may be students on this webinar. So as I already mentioned, we've got cohorts in place where we identify potential donors for diseases of interest. We get blood samples for them because we are looking at human immune responses, and we have systems in place from where we then isolate B cells to make the monoclonals. So when we started doing this work, we started off with very traditional ways. We had traditional restriction-based cloning methods in place, and when we would make monoclonals, this entire process would take several months in a good way, and sometimes over years to make the first set of 100 monoclonals. And it was obviously a very time-consuming process. So from there, if you would click again, we then got technology to then do recombinant based cloning, because this is a single cell cloning that we do. So it takes a lot of steps, and this could be pipe cloning or Gibson cloning. And we then collapsed this timeline to about a few months. And now where we are, which is the last part of the slide to the right hand side, is that we have a lot more high throughput methods in place, where we either do repertoire sequencing or high throughput recombination based sequencing. And essentially what happens is that we've collapsed all this into a few months. So the discovery that we do takes a lot for the characterization of the patients, and then of course the screening of all the monoclonals. But all the work in the middle, which involves a lot of cloning and expression, has become very, very high throughput, not just in my lab, but multiple other DBT labs across the country. So can I please have the next slide? So now what I'll do from here on, I'll transition to telling you a few things that we have at ICGB right now in my lab. So at ICGB in my lab right now, we have monoclonals on dengue, chikungunya, SARS-CoV-2, we had made a few. And then we've just embarked on Chandipura virus as a part of the DBT iBricks PhD program. And each one of these programs has been something that DBT has very generously supported, and through NBM and BIRAC. So please, the next slide. So what I'll do is I'll start with dengue, because that has been the largest program in the lab for a while. And there are multiple DBT supported small projects and large projects that we have, and BIRAC primarily supported this, where what we did was we got blood samples from patients that either had acute febrile infection and then followed them out till recovery. And then if you see this little cartoon that I'm showing here in the middle of the slide, we got effector B cells from them, we got memory B cells from them, we did our single cell sorts, and we made almost more than 400 monoclonals. Then all these 400 monoclonals got screened for their binding ability to the virus and their neutralizing ability. And if you know dengue virus, most of you would recognize that there are 4 serotypes that are known of the virus. And the goal of this entire BIRAC-funded investment was to find monoclonal antibodies that neutralize all 4 viruses to equal potency. The importance of this is, if you get an infection, you cannot predict which serotype would you get it. And so whatever therapeutic or prophylactic option that you come up with has to be such that works across all 4 serotypes at the same time. So we found a lot of interesting monoclonals, and this is not something that we are going into basic biology, but I wanted to just flash three graphs here that we found multiple monoclonals that we've now patented, which neutralize all four serotypes of the virus. And not only do they neutralize all four serotypes of the reference strains of the virus that we use in the lab, but these also neutralize circulating strains in India. This point is important because you see the reference strains that we typically use in labs are strains that have been isolated in the 1960s and the 1970s. And as we've learned through COVID, virus evolution happens. So, what we will see as of today circulating, even for the most stable viruses, may look very different from reference strains. So, these antibodies actually also neutralize circulating strains. So, that has been a fantastic finding. Can I please have the next slide? So for chikungunya, we did something similar. We again had a project supported from DBT to look at human immunity to chikungunya because it causes a lot of debilitating arthritis. And what we did there was individuals that recovered from chikungunya, which is a primary infection, we baited for memory B cells. So if you follow this FACS plot out, the last one shows you memory B cells where we've actually used virus and we've pulled out virus-specific memory B cells. And then we generated about 200 odd monoclonals, and we did a smaller amount here because we were starting from virus specific B cells, so we knew we didn't have to do too much screening. And then if you would click again for me, please. Thank you. So we found two monoclonals that are fantastic. They are, if you look at the plaque assay that I put here, you'll appreciate it. And then if you follow the graph out that I'm showing, these two monoclonals turned out to be extremely potent. Their IC50s are in like 0.0003 micrograms per ml, which is absolutely great. And chikungunya actually has a mouse model that one can use. It's a very simple mouse model where you inject the virus in the footpad of the mouse, and then you can gauge the swelling that the footpad has. And this is a direct correlation of the kind of joint swelling that humans see when you get the acute infection. So we recently also tested these monoclonals in vivo right at ICGEB. And what we've actually found is that both these monoclonals work both prophylactically and therapeutically, and they bring down morbidity and the joint swelling of the footpad of the mouse. So they do work very well in vivo. So we are hoping that we can move these forward and stabilize the clones towards biomanufacturing. Can I have the next slide? And this, I think, would be my last data slide. So this last data slide, I think people at DBT may have seen before. This is the SARS-CoV-2 work that we had done. One of the major things that was coming out that the virus was mutating rather quickly and we were seeing all the variants. So one of the things we were trying to do is find monoclonal antibodies that could neutralize broadly across multiple variants, and that's what we found. After we made monoclonal antibodies from recovered individuals that had actually seen the Wuhan virus, we found an antibody that actually did not block the ACE2 RBD interaction but bound from behind the RBD. So it did not allow the RBD to open up to bind to the ACE2 receptor. So that essentially made it really broadly neutralizing because that was a very conserved space of the virus. So if you would just click so that the slide is visible completely. So we did publish this, and there are 4 antibodies that now BIRAC is supporting, that we bring a cocktail of these antibodies together, because as we moved through the COVID era and we saw other companies working the sphere of monoclonals, one of the lessons we learned was that one antibody may not be sufficient for a virus that evolves very, very quickly. So you want to target the virus from multiple directions so that you can neutralize the virus and you don't get a lot of viral escape or immune escape. So that's the entire premise that BIRAC has requested us, that start with 4, see a cocktail that works, that actually increases the potency of the cocktail and also stabilize the clones and move them to biomanufacturing. And these are the monoclonals that we are moving forward towards that right-hand side that I had started with, towards the development of monoclonals. Next slide, please. So this brings me to the last slide, and I hope that this webinar actually becomes a discussion right here. So if you would just click on the first part. Thank you. So one of the things I was thinking about when I was putting these slides and DBT kindly invited me to participate in the webinar was that I come from the left side of this graph. I'm an academician and I can spend hundreds of years looking at single monoclonals. I will keep taking monoclonals, and there are so many things, such beautiful immunology that one can do of looking at their structures, their function, their understanding, correlating it back to what was happening in the human being. So the left side of this graph is where I can spend my entire career on. But now with all the kind of initiatives that DBT is taking, I think that, and then there's industry that's on the right side of this graph. And then I did not make this graph. I actually found it on in a wonderful review that I have actually pasted on this slide at the right hand side bottom corner. If anyone here is interested, it would be fantastic if you go and read it. It was actually titled as Lost in Translation: The Valley of Death Across Preclinical, where I stand, and towards clinical side. But if you could please go back to the previous slide. So thank you. So my interest would be, without actually reading the bullet points, would be that how can I meet midway? That people like me, not just me, but so many of my colleagues who are in the academia space and may have things that we could move in the market in India and indigenously make, that we could move biomanufacturing further towards novel molecules that we discover, and something that Dr. Vaishali had alluded to of availability, accessibility, and affordability is a gap that we can bridge. And the points that I would think about is that people like me who can spend a lifetime doing this work, what is the minimum data that is required? What is the kind of rigor that one would need to do it? And of course, the other two bullet points of, just engagement is something that we are already doing with this webinar. So thank you so much for it. And with that, I would stop and thank you. And I have an acknowledgement slide because obviously I don't do the work. I just come and speak about it. Thank you.
Thank you, Dr. Chandil, for providing an excellent overview of the monoclonal antibodies subsector from an academic perspective. Now, I would like to invite Dr. Vinay Ponaji, Navya Therapeutics Private Limited, to present the industrial perspective. Dr. Vinay Ponaji is founder and director at Navya Therapeutics Private Limited, Hubli, a startup focused on targeted therapies for oncology and AI disorders since 2023-24. He also served as managing director and BU head at Shilpa Biologicals Private Limited between 2018-23, and prior to that, he was the founder and director at Navya Biologicals Private Limited Hubli between 2007 to 2017, a startup which focused on development of pipeline of biosimilars, RHSA, and a continuous manufacturing platform. With this, I invite Dr. Vinay.
Thanks a lot, Dr. Yadav. So pleasure being here. Thank you for inviting me. I'd like to take off from where Dr. Anmol left. So I'm on the right side of that graph that she showed up, which is where we essentially like to be. Can you take it to the next slide, please? Next slide. So I'd like this to be a little more of a discussion rather than me giving some yarn for 5-10 minutes, and after that, we'd like to hear a lot of questions on this. So on the right side, the market for biologics, monoclonal antibodies is likely to be about the $700 billion in the next 8 to 10 years time frame. A lot of it is actually going to be used from hospitals. And the bad or the good part, depending on which side of the equation you are, India and Asia is going to be the fastest growing market for these. I'd not like to see this, but from a medical perspective, that's not a great thing. But from a manufacturer, from a research perspective, that's a good thing because there's going to be a lot of demand for monoclonal antibodies. Next slide, please. The next slide, please. Yeah. Strangely enough, so I'm going to break down my talk into 3 parts. One part is novel drugs, the second part is biosimilars, and the third is there are new modalities of treatment that are coming up. And in all these modalities of treatment, new biologics, biosimilars, etc., there are some trends which are overlapping, and that we'd like to identify. And this is something that most people embarking on a monoclonal antibody development and commercialization journey need to keep in mind. Speed is the highest. Speed gives us the biggest advantage. So if you look at any market, for novel drugs, the first two people to release antibodies for any new target end up capturing about 70 to 80% of the market. And then there are Me Too biologics or Me Too mAbs, which come into novel mAbs that come into the market, and these guys are fighting for the crumbs. So at the end of it, we need to remember a couple of things. Time to market, how quickly we get into the market for an unmet need is extremely important. That means the discovery part, the scale up, the clinical studies, and the time getting into the market, the manufacturing part, all need to work in sync for a product to get to the market from zero to hero. And that typically now involves the COVID has taught a lot of lessons, and that timeline has been crunched from 7 to 9 years to about 2 to 3 years. And that's something that we need to keep in mind when we're looking to make India a superpower in monoclonal antibodies. So there are two components. One part is the technical part. The second part is also the buyer and the payer part. This is something that we need to keep in mind again. At the other end of the spectrum, there are buyers, and typically people coming in later end up targeting smaller markets, and they end up giving bigger and bigger discounts to either the buyers or to the retailers or the distributors. But that is never going to make up a kind of margins that these guys would have getting from first two or first products in the market. So no matter what you do, if you are the fourth, fifth, and sixth manufacturer of a particular drug, it's to take back the money that you have invested. So that's a takeaway for us from this particular slide. Next slide, please. Next slide, please. No, the one before this. Okay, can you go before this, please? So now to buyers, India wants to dominate the market. Unfortunately, we do not dominate the market. There's the price in India. It's 2 to 4% price. 1850, $1860. The market cap of the Australian app is the of the market price. I think back again. Yeah. So if people are selling at $2,000, $2,500 with US market, although prices slightly high, we are not at this point in time. This kind of the market, how are we going to be profitable? That's something that we need to focus on. The people are going to be developing drugs, and that sort of the brain, so, given all this, what are we going to do next? How are we going to ensure that we are going to be successful? We're going to be able to sort of take these drugs to the market. Next slide, please. So this is a contextual observation. India has been a superpower when it comes to genetics. Clearly,
Dr. Vinay, I think there is an internet issue from your side. Dr. Vinay, I think there is an internet issue from your side. If you can switch off your camera, I think the voice will be more clear.
Am I audible now?
Yeah.
Yeah. So contextual observation. India has been a superpower when it comes to generics because of its process engineering and synthetic chemistry skills. Combined with the fact that the pathway for generics is harmonized across the globe, that is superpower here. Unless similar strategy falls in place or circumstances fall in place, our ability to become a superpower in monoclonal antibody production or in antibodies is going to fall short of the expectations that we have. And we really need to on this, both from and as well as from regulatory and ensure that at the same time advantages companies and for the last couple of decades. Next, all is that after the market, the more people it is companies across we and for that all these businesses need to come together technology everywhere. Yeah, so 80% of the cost typically goes away in the clinical trial part. Now, this is the part that is going to take up the largest amount of time for all commercialization companies. And this harmonized scale across geographies. This would be a rate limiting step, and which should be build up phase one studies so that the timelines to the market are shortened. Phase one study capacities, if we can carry the full power study, can be relevant across the globe. The phase three trials, depending on different geographies, different requirements, but when it comes to phase one, we really need to focus here. When it comes to biosimilars, the biosimilars now be approved based on phase one equivalence, and then in some markets where phase three is required, can really take those steps through there. And the greatest the shift needs to be in terms of the early physical clinical characterization of the product, something that Anmol was alluding to in her presentation when she was doing it. Next, please. Development. Most of the time, I see that animal models which are not really relevant are being used for so-called application for phase one or phase three. This is not really required. We are seeing introduction of organ-on-chip technologies as an add-on tool, so that could be used. And if we really have access to animal models, we should be focusing on short term because only wasting a lot of money. The that most of us to do science, but we don't want to record things that are happening. Challenge for all of us. Unless there is traceability of data, unless we record everything on time, you will spend developing a product and then will not get into the markets and data to prove that you've developed the drug in a particular way or some critical data is missing, or people would term it as fictitious. So we need to be really careful here. We need to bring in electronic systems of tracking and managing data as early as is feasible. The last part is that the line between R&D and manufacturing is blurring. and we need to be cognizant of this. So people starting out on a project is really encouraging. You can almost be sure 5 to 7 years people doing it at large scale will be the ones who are also going to be producing it using our tech production, and that's something that really is going to happen in the next 5 to 7 years. It's happening, but next 7 years to see this. Next slide, please. For people who are setting up manufacturing units, environmental regulations need to wasted on trying to create the next environment. MABs essentially use BSL1 or BSL2 technologies that need for us to really have wait for 1.5 to 2 years, which is now being addressed and as we speak. Now, when we get stuck on manufacturing and over the years, next slide. Next slide.
Dr. Konaji, can you see the slide?
Sujit?
Yes.
No, it's stuck on the setup challenges for maps. Yeah.
Regulatory challenge. Can you see that?
Long duration for approvals. Yes, I can see. So the long duration for approval, the facility and then the molecules. This is something that the CDSC is now trying to try and fix the timelines, but there's a lot of companies critical time to market. A great decision always this CDMO for people developing their own molecules. This is being signed by these people in efforts to streamline this because this is going to become a challenge if we are to become a powerful global. Next. Next slide, please. Sujit, can you hear me?
Yeah, I can hear. Can you see the slides? Is it an evolution of monoclonal antibody manufacturing?
No, yeah, it has come up. So,
Dr. Konaji, can you hear us?
less energy efficient or less viable than the fed batch processes. For the last few years, the downstream side has been significantly debottlenecked also with the advent of SMB and charge filtration systems. This has really brought down a continuous process can actually be a continuous process. There is no holding of the material in between. And we get the product right from the clone stage to plant all within a span of around 30 days to produce the same amount of product at 3 or 4 batches of fed food. So this has significantly brought down the cost and the footprint. The footprint contributes about 60% of the cost of the monoclonal production cost, and that has been brought down significantly. And this is an area that we need to be really focusing on. The reason for this, next slide, please. As the next slide comes up, you would see that there are a lot of pack tools and technologies, bioprocess technologies that have evolved right from the filtration system to the charge filters to assemblies. that allow for continuous processing of monoclonal antibodies. That's what you see on the right-hand side, as well as the process technologies. These are the technologies that enable us to move from lab scale to production very, very quickly. And the change in volumes are not so very different lab and manufacturing currently. So, for those of you who are looking to actually produce from a lab scale kind of product, as long as you're certified as GM, it would be possible to produce at almost the same scale as the lab scale production systems. Next slide. So, as the slide comes, let me continue. There are two factors here. One, we have highly fragmented markets, different modalities of treatment for the same target. So there could be a T cell therapy, the slide before this. There could be T cell therapy, there could be MABs, there could be NK cells, etc., etc. So that is breaking your market into smaller and smaller fragments. At the same time, there's going to be immense competition. Given all this, how are we going to ensure that we still retain leadership position? How are we going to ensure that we are still going to be viable despite all the investment that goes into setting up a manufacturing unit and a GMP manufacturing unit? Now, these challenges that often face, and the only way that we do this is by adopting continuous processing is for monoclonal antibodies. So, lab scale, essentially, a lab also be a production scale process, but without and the not have capital expense that it would drain you completely. So, as an example, we spent about 250 for putting up a monoclonal antibody facility based on a patch. And if you had to get from a continuous process unit, if you had created a continuous process unit, the amount of capital that would go in would be 80 to 100 crores, where you already saved costs of setting a monoclonal facility. And also, that you can only focus on when the need arises. For example, Dr. Anmol mentioned that she's looking at chikungunya or she's looking at dengue. These are seasonal requirements. So if she does whenever she comes up with the tests for these, you're going to have seasonal requirements. Unless we have something like a processing facility, we are not going to be able to meet the demand. But at the same time, the demand disappears almost overnight, and we should be in a position to take on other challenges, other MABs to produce at a lower cost, and that can be only done with a continuous facility. So the merging of R&D with manufacturing is going to happen. That's the exciting part for all of us who are in this webinar today. The next one, please. And the wider adoption by the government procurement programs is only going to happen when the cost of goods falls below $50 per gram. This is a challenge for all of us. I think that all of us really need to work on this. If monoclonal antibody for dengue or chikungunya or any other infectious diseases needs to be financially viable, we need to be able to produce it at lower than $50 per gram. How are we going to do this? That's the challenge. And I invite questions after this. We'd love to see a discussion on these aspects. So thanks a lot for hearing me out.
Thank you, Dr. Vinay Konaji, for providing an overview of the monoclonal antibody subsector from an industrial perspective. We will now open the floor for questions from our audience. Please type in your questions in the chat box, and our team of moderators from DBT and BIRAC will assist in facilitating the discussion. Over to Dr. Amit Tripathi from DBT and Dr. Sujit from BIRAC.
So,
Amit, we can't hear you.
no, but Shivani can hear you.
Yeah.
Sujit, maybe you can start.
Sorry.
Till Amit sorts out.
Am I audible? Yeah.
Yeah.
So the question is from Dr. Richa to Dr. Chandele. She is asking, how do you think the increasing focus on personalized medicine will impact the development and applications of monoclonal antibodies?
Thank you so much for the question. From what I understand, the question basically is that how would you, what exactly is the question, that how would precision medicine help? So I think this place where monoclonal antibodies, at least India should be focusing on, are diseases that are of public health importance to India. So, and also diseases where vaccines are not universally available. So that's how we should look at monoclonals. We should not continuously keep. I understand they are always considered as precision therapeutics, and that's primarily for the space of cancers or specific things that one speaks of. But when it comes to infectious disease monoclonal antibodies, one needs to think of them as just another method with which we can cull infection and disease. So, does that kind of answer the question of how they would play a role, or am I misunderstanding the question?
Yeah, so that answers the question, I think. So there is another question from Dr. Prachi to Dr. Konaje. She is asking, what are the main challenges in scaling up monoclonal antibody production?
Consistency, number one, and enough productivity. Does the process have enough productivity? That's number two. And the last part is we tend to overlook this, but most of the time we do not note down events that lead to high productivity or high quality, and that's something we need to keep an eye on. This is the challenge that we face. People do not know, they not observe why really one batch of batches produce really high and the other ones don't. We don't analyze. So it's more of a mechanical kind of a thing that happens where you produce batch and at the end of it, you're only looking at the quality of the product, but you're not really looking at the productivity and the circumstances that lead to higher productivity or better quality. This is something that we only need to have.
So may I add one thing to Dr. Vinay, that from the basic biology side, one of the things, like for example, the way my lab is making monoclonals right now is transient transfection. Even though it's a fantastic system and we are using the best reagents possible and we get very good yields for what we want to do, the key step from there is that if someone like Dr. Vinay was to receive a clone from my lab, we have to bridge the gap that we give him a clone that is stable so that every time he goes from batch to batch, that does not change. And that's what he's talking about, that becomes a little stochastic. So when I go from a transient to a stable cell line, where exactly the gene will integrate will again dictate how well does that gene express as protein, a protein that does not aggregate, a protein that meets all GMP guidelines, that Dr. Vinay can then move forward as a therapeutic. So that entire process that I've just stated on one thing takes a lot of effort, and as he was saying, a lot of precision. There's something that works really, really well in my lab and is a very good expressor. As soon as I stabilize it, it may just lose its expression to the same level and it's of no use, however potent it is.
Yeah, but the good thing about all this is that value of death that Dr. Anmol had showed up in the graph, that is becoming a little shallower with people adopting processes that are very similar between the lab and the manufacturing setup. So that valley was very steep maybe 3 4 years back, but with back tools coming in the picture, that valley is getting a little shallower, and therefore don't see as many deaths these days as there earlier.
Yes, Dr. Vinay, I completely agree with you. As reagents are getting better, once upon a time, the entire time used to be spent in the go and no go of whether the protein was even aggregating. Now we don't face those kind of challenges. I completely agree. Every year is a better year with better tools and technologies.
Yeah.
Yeah.
Okay.
So,
Thanks, Dr.
in fact, from production perspective,
Please go ahead.
yeah, yeah, I added one point. From our perspective, manufacturing plants should be during the correct product 99% of the time, and that's typically the benchmark that is coming in place now in India.
Thanks, Dr. Vinay and Dr. Chandani. So there is another question from Dr. Gautam for Dr. Anmol. He is asking, what key technological advancements do you foresee shaping the future of monoclonal antibodies in the next 5 years?
So I think we just made a huge leap. I think in my second or third slide, I quickly breezed through it. But previously, even prior to the pandemic, making monoclonals wasn't something that everyone was doing or could do. I think with the pandemic, what happened is that so much development happened really quickly, even in the sphere of discovery, that the lag time that used to be at the discovery stage, where a needle in a haystack approach is what one would take, first and foremost, that I would like to acknowledge is that just the discovery time has completely collapsed. So, if you look at some certain labs in the US, for example, they are funded to do races. Like in 29 days, they will go from no monoclonal to an entire fully functional human monoclonal. So, I think that itself, we should take a few minutes and rejoice that if we were to have like all those robotics and everything in place in Indian labs, our discovery that we take still take a few months to perhaps a year, the labs are collapsing it to a month. And I look at it as like the 4-minute mile, that once upon a time human beings could not run the 4-minute mile, and one day one person ran it, and now people are breaking records all the time. So we've already done that. I think uh, more than just making things, revolutionizing things, I think accessibility to such technologies and tools and they becoming more ubiquitous. That's something that my lab or my colleague's lab is not working on. There is another scientist who can work on it, would be great. The second thing is, I think, apart from discovery, where we need to make key strides is the translation. I think if we could make strides in, right now we are still relying on small animal models. I think where the discovery will happen is if they are organoid models or something that will really speed up the process. So once we have a candidate, I'm now looking for, oh, what's the best animal model? And if it's something like dengue that does not have a great animal model, the best animal model is an NHP that does not show disease. At the most, it may show a rash. Where do I go? So, if we had in vitro systems that could kind of mimic something that a mouse or a hamster or a NHP doesn't give me, that's something that I'm really looking forward to someone doing that magic in innovation.
Dr. Anmol, in continuation to that, just I'd like to add one more question is there from I'll take from one of our participants, Dr. Dilip. He's asking the production of monoclonal antibodies is expensive. So due to this complex manufacturing process, like or lengthy development timelines and the need for specialized equipments and facilities. So reducing the production cost while maintaining a product quality is an ongoing challenge. So what can be done in this particular direction?
I think India is a fantastic example of affordable healthcare. We've shown it over and over again that things that are expensive in the rest of the world, and I'm not a person who deals with the economy, but I think there's a lot that gets added when you manufacture something which adds to the cost. I think if we want to make things affordable, as we've done in multiple different sectors, including space, we should be able to do it in biotechnology, that when innovation and manufacturing is in our hands for problems that we have to deal with, we always do them in an affordable way. So that's where the innovation comes in. If we keep relying on patented technology, patented media, patented everything, that's where the expense increases.
Thank you. Thank you.
So I'll add on to this a little bit here. See, if you were to consider the cost of MAB at around 100 rupees today, 60 to 70 rupees is coming from the facility charges cost itself. So if I were to simply bring down the size of the facility to half of what it is today or one third of what it is today, you already brought down the cost by a significant 35 to 40%. So that's number one. And number two, you have packed tools coming in, adopt those packed tools, you get more consistency. You're in a position to bring down the cost of your process a lot more. So I would expect the low-hanging fruit to bring down, which is the size of the facility and the packed tools adoption, to bring down the cost of MABs by at least 50 to 60% from what it is today. Mind you, as compared to global standards, we're selling our biosimilars at between 2 and 3% of the costs in the US. Those may be misleading, but 2 to 3% of the costs in the US today, if you were to bring it down to half of that, we're going to be able to increasingly have a lot more people adopt monoclonal antibodies as their preferred therapies.
So, due to time, thank you, Dr. Vinay and Dr. Anmol. Due to time constraint, I would like to request the participant to please send us your queries in a post feedback mail which you receive after this webinar. So, we'd like to address the remaining questions on that. So, we'll just take one final question for Dr. Anmol. From Dr. Fernandez from Hyderabad. The question is, ma'am, the need for high quality congested raw materials such as cell culture media, bioreactor components, or chromatography resins is essential for successful monoclonal antibody production. So shortages or variability in these raw materials could sometimes affect the production schedules or overall supply chain. So how can this be dealt with in the long run?
I think we kind of answered that question in a way. Dr. Vinay answered that question in the last question itself. It's already dovetailed there. But I think it's going in parallel. We are getting better. We are not relying on a lot of imported things. DBT is actually making a lot of effort to, help so many startups. I think as innovation happens in all other sectors, tomorrow I may not need to go to a fancy company from another country to import a media or import a column to do the work we do. So I'll keep it short, but I think it all comes down to innovation, not just me making monoclonals, but everything else that you just listed. It's a fantastic question.
Thank you, ma'am. I think we are done with today's Q&A session. Really thankful to the experts. I'd like to hand it over to Dr. Amit, please.
Thank you, Dr. Tripathi and Dr. Das. As we come close to the close of this insightful webinar, I would like to extend our heartfelt thanks to all our speakers for their valuable contributions. Your insights have shed light on the immense potential of this subsector of biomanufacturing of monoclonal antibodies. For those who are unable to attend live, the webinar recording will soon be available on the DBT and Bioreactor websites. Finally, please mark your calendar for our next webinar in this series, which will continue to explore various thematic areas of the Biofoundry and Biomanufacturing initiative. Details will be shared soon on the DBT and Bioreactor websites and through various social media platforms. Thank you once again to our speakers, participants, and the organizing team for making this event a success. We look forward to your continued engagement in the upcoming sessions. Thank you. Thank you.
Thanks a lot, everybody.
Thank you.
Thank you so much for having us. Thank you.
Thank you so much
Thank
for
you.
joining us.
Thank you.
Bye.
Thank you.
Thank you, ma'am.